Okay, so I have this slide, and I do want to go through it with you guys because it'll, I hope, tie a few things together for you. Then, if you want, and not if you don't, my feelings won't be hurt. I have perhaps an alternative presentation, just very simply introducing the ideas behind model evaluation, which Mona went through, but I think we were kind of rushing those that day. So you guys can tell me if you want me to go through this or not. It's only 232 slides. It's not 232 slides. Okay, so this is just a kind of a flow chart. You guys will have access to this, so don't worry about it. But this is just intended to kind of sum up how this stuff works, at least in my brain. And I think if you went to Mona or Cassandra or Xiao, you might have a different view. And if you go to a different lab tradition, you might get a very different view, right? Because in some sense, we're a little bit inbred. Mona is academic offspring, and I'll claim Xiao and Cassandra as academic grandchildren. So in some sense, we're a little bit inbred here, but you signed up for the course, so live with it, right? So if you want kind of a base for reading, you can go to a book that we published in 2011, and that gives you a review of the kind of the conceptual basis in chapters 2 and 3. There you go. And then it has a middle set of chapters on kind of methodology, which is getting out of date very quickly. And then it has a final section of chapters on applications, which is completely out of date. And I begged the editor to let us not do that section, and he said, do it. So what I've done is I've kind of assumed that you've got that knowledge, and you've been exposed to a fair amount of it this course. And then I've added in key papers that you should also read that are relevant about each of these steps. And in typical academic conceit, most of them are written by my group of people, so put up with it, okay? But I do want to take you through this and kind of tie all these pieces together for you. We're going to start in two places. Here's where we're getting our environmental data, and up there is where we're getting our occurrence data. There we go. So we talked about occurrence data and where you can get them. And then we talked about some things of looking at data quality and data precision, okay? And I'm just assuming that you look very carefully at the quality of your data, and that you look for outliers, and that you get in and you be good analytical scientists and you just explore those data. You look at them in geographic dimensions, you look at them in environmental dimensions, you check the outliers, and you make sure that they are real and not erroneous, et cetera, et cetera. All the things that you should do as a matter of course. And then we've also talked about getting your environmental data, and we've talked briefly about reducing dimensionality. My current guess is that the best thing to do is just to get rid of redundant dimensions, which is to say look for correlations and pick some criterion of high correlation and eliminate one of each pair that shows high correlations. Okay, so we're kind of through this very initial data phase. Then we have to kind of these 3 or 4 boxes take us into things that are a bit more complex. One is that the environmental data may be able to be reinterpreted to create data layers that are more directly relevant to your species. So for example, you might have temperature data, but botanists love growing degree days, right? And that may be extremely relevant to your organism, to your question. And so there may be a step of reinterpreting, combining, integrating environmental data sets so that you create data layers that are kind of maximally informative for your taxon. Okay? It's pretty crucial to look at the amount of spatial autocorrelation. We didn't talk about this much, but this is a kind of a complex interplay of how big a study area is, what is the grain or spatial resolution of the environmental data, but also what is the underlying density and grain of the information that was used to produce the environmental data. And we all talk about WorldClim. I personally hate WorldClim. If you look at the map of the weather stations that went into WorldClim, some regions have incredibly dense points of weather stations, and then a neighboring region may have nothing. Okay? And so those regions where you have dense concentrations of weather stations are going to have more detail. And those regions where you have no points are going to be very broad and interpolated. So you have to look at the spatial autocorrelation, essentially how smooth those environmental data are. And you have to remember that that may make points non-independent. Right? If your environmental data are very smooth and very highly lagged in terms of their autocorrelation, then 2 points that are 100 kilometers apart may not be independent. So you need to think about that. Again, we didn't talk a lot about that in this course. So we've kind of refined a bit in our view of the environmental data. You may have to throw out some point data because of that spatial autocorrelation. But then we get over here, and you should note that M is the center of the universe, right? And of course it was ignored absolutely until 2011. Read this paper by Barve. This is this niche modeling working group that kind of started in the period where Mona was just leaving. Were you on that first paper with Alberto? Maybe you had already left. I'm sorry, forget it. Keep talking, don't worry about it. Okay. So no, I know you're not on that one. This is a group of people at KU that's been working for 12 years now. We meet weekly. We do projects together. It's nobody's thesis and it's nobody's class. They're just projects, and they get done collaboratively. And so you're going to see these kind of ridiculous lists of names. I think I abbreviated them all because we'll have 10 or 12 authors, but it's honest that these are all people who contributed to these papers. So one of the very first ones was this Barve et al. Paper. And this was pointing out, first of all, that the calibration area matters in everything that we do in niche modeling. And second of all, that we could reasonably equate that calibration area with M. And it even gives kind of a caricature of how you might estimate M. And now, 7-8 years later, we're coming back to that. And I'm hoping in the next year we'll have some software that will implement that fully. So this is kind of a crucial paper. because it's essentially saying this is the area over which you're going to calibrate your models. Another piece of this, which we've mentioned slightly briefly, is assessing the BAM scenario for the species in question. You remember that first day, I put up all sorts of different topologies of the rings of the BAM diagram. And what the Saup et al. Paper says is that those topologies of pairs or trios of rings determine whether your effort to fit a robust niche model will be successful or a complete failure. And so essentially we talked very briefly about Wallacean species, right? Wallacean species are essentially impossible to do a good niche model for, whereas Hutchinsonian species are quite feasible. So you kind of have to think about, at the extent and at the grain that I'm interested in working, what does my BAM diagram look like? Which is to say, are there accessible sites that are not used by the species? Or is everything accessible also suitable? That's really what it boils down to. But that's this paper by Saup. Okay, so by the end of this, we've determined whether we've got the right calibration area and the right grain and resolution in our study. So then we take this little jump over to this box here, estimate ecological niche, various algorithms. This is the calibration stage. And it's this tiny box, but as you saw yesterday, it's a lot of work and thinking. And so, we then have this two-headed arrow where we may go back and forth with different parameterizations and different possible calibrations. And so this was introduced by Warren and Seifert, 2011. And you saw Marlon had some neat ideas about how to make that more feasible. So we get to the end of that calibration step, and you remember the latter part of what Marlon was doing, even though I asked him not to, was to transfer the model to, in that case it was future. But essentially this is just after you've done the correct parameterization, you want to transfer that model to geographic space. And so now we have a map, okay? And here's where we go into this loop of evaluation and thresholding, and maybe the order's wrong there, maybe it's not. But essentially we need to ask, after we've done our calibration, do we have a map that has significant predictive power? And again, if you guys want my 632 slides or whatever the number I said was, we can talk about that loop in just a moment, okay? But let's imagine that evaluation is successful, and this is a paper that Mona was on, so that was before she departed my lab, where we proposed the partial rock approaches, and Mona was also on this one where we pointed out a bunch of details about overfitting and model transfers. So let's imagine we pass this test and our model indeed can be challenged with independent data and yield significant predictions, which is to say predictions better than random expectations. Okay? So now we have a model that's not only a map, but also it's passed the test. So now we can start interpreting. And of course now we get into realms where we have different uses for this model. We may be interested in current distributions. We may be interested in some model transfer to the future or to another continent. And so that's where we have to ask about whether that transfer to another time or another place is not extrapolative. And so we talked about MESS and MOP. Please use MOP, not because I want you to cite my paper, but because it's the right conceptual framework for asking questions about extrapolation. So you've got this Owens et al. Paper for that. And actually, I thought I'd just added this. It's weird. Huh. Well, there's a paper by Huijie Qiao and colleagues on this idea of using various algorithms. It's the infamous no silver bullets in niche modeling paper, and makes for fun slide presentations because you can put up pictures of werewolves and things like that. I'll add that in before I put this presentation in the common stash for you all. But essentially, when you're going to do these transfers to another place or another time, you have to make sure you're not doing extrapolation that's out of range and shouldn't be interpreted. And then you also have to make sure you're not doing extrapolation that's within range but to weird combinations of variables. And this kind of speaks back to what Xiao just told you about with these changes in collinearity or changes in correlation structure. So I can very easily imagine adding a box to this diagram where we'd not only assess extrapolation, which is essentially how weird are the conditions in the region to which I want to transfer my model, given the conditions where I've calibrated my model. But also, this other box would be how weird is the correlation structure in my transfer region compared to my reference region. If those correlations switch around or disappear or appear, then I may get much worse results as a result of doing that transfer. And so in here is where you do your interpretation and you get your science insights. But that's essentially pretty much of a view of the whole process. So questions about that? What I've tried to do is kind of get it all on one page for you. I rarely take 10, 15 minutes to explain one slide.
Is there a way to make a post-fit assessment of autocorrelation and how it's impacting your response as you would do like in a general linear model framework, that you look at the residues and see if there's problems or then probably, I don't know, the maximum entropy versus the maximum likelihood might have some differences that I'm not seeing yet. So how you treat after you fitted everything, like an equivalent of looking at model residuals.
Yeah. The closest I can give you that is that when you're in here, if you think about model failures, remember we're dealing with two kinds of autocorrelation. One is kind of how smooth or how detailed the environmental data are, and the other is how clumped are the points, right? But when you're in here and you have a model evaluation, remember we've talked about this E parameter, which is what proportion of the points we're kind of okay with being left out. Just as you inspect residuals for kind of a random distribution or lack of pattern, you really want those left out points to also be fairly randomly distributed. So one signal of a problem when you're thresholding is, you do that threshold where you say, I'm okay with 5% of my points being left out. You want those 5% to be kind of scattered. But if all those 5% are up in one region, you probably have a problem with your balancing of the points or your model thresholding choice, or something's wrong. And so we see this we've seen this very recently where we're doing these models of ticks that are vectors of Lyme disease and other pathogens in Eastern North America. And what you see is huge amounts of monitoring kind of in Wisconsin, Minnesota, and the Northeast, because it's where they have Lyme disease. And that tick is in really high densities. You come to Kansas, and that tick is present at really low densities. And it transmits Lyme disease, and none of the doctors know about it. You do the models, and the whole the whole Eastern Great Plains gets left out. And it's because you have such density of sampling in the east and the north, it's really hard to rarefy those data and balance the sampling. And so, the CDC published a niche model that gave the leading edge of the distribution of Ixodes scapularis as being in Illinois, even though we have it in Kansas. And, so their model was just wrong. But essentially those residuals, those points that were far off of the relationship, I'm pulling hard on the analogy to make it work, but those residuals were clumped. And that was a serious sign of an error that they missed. And so they published a map that was pretty misleading. Should have thought of that one for the bad paper shooting gallery.
Hello, there you go. The idea of filtering down clump data for species like ticks or other things that have monitoring programs, seems like there would be a way to develop an index where you base occurrences on sampling effort and use the index value on whether or not as a threshold to include it in the model. And I could see someone who's good at developing indices might want to take that on, but because there are a lot, I'm from the field of entomology, so there are a lot of programs to monitor invasive species, and they're usually on the state level. And so you could look, say, at the CAPS program and see how rigorous the sampling effort is in that state for X species, if you want to model it, and then develop your index to threshold out those clumped distributions.
Yeah, and in fact, we didn't talk about it a lot. Mona mentioned it when she was taking us on the grand tour of Maxent. But Maxent has the potential for using a bias surface. And essentially, imagine that you go to each state or even each county and get a total number of hours spent flagging for ticks or total number of trap nights for mosquito traps or whatever. You can put that in as a bias layer to Maxent, and then that background will be sampled from that distribution, essentially weighting the areas that were heavily sampled more than the areas that were lightly sampled. That's not a very common situation. It's usually, medical entomology and a few other situations. Another way of getting that bias surface is to take a reference group. So let's say I'm studying, one bird, one songbird, and I'm interested in what was the sampling process that led to those 52 records that I have of that species. Well, I could also take all of the reports of that order or that, or of all birds, some reference group that I can justify as having been sampled similarly to how my species was sampled. Well, you back down to something that doesn't have biogeographic limitations across your study area, and you basically just say, if an observer was there, he or she would have reported one or more of these species in my reference group. And I'm going to assume that that's an index to their opportunity to have detected the species that I care about. Okay? So for example, my student who's looking at albatross distributions in the southern oceans, she got her, X number of thousands of records of wandering albatross, but then from the same source, she got all Procellariiform data, so all tube nose data, that order that includes albatrosses. And basically, her, I think, very reasonable supposition was anybody who is reporting data, recording and reporting data that include tube noses, so it's basically offshore, high southern latitude observers, would be reporting the tube noses and especially would be reporting wandering albatross, which is the coolest of the tube noses, so they probably would report it. And so that seems to be a reasonable focal group or index group for that. Now those bias surfaces, I've experimented a fair amount with them in Maxent. And if the bias is huge, then what it tends to do is the less sampled but not zero sampled areas basically get pushed to zero, and you get no sampling of them. And so, we've played with doing things like a log transform on the survey effort if there are big disparities. And that kind of works, kind of doesn't. And then in too many cases, you just can't in good conscience posit either a means of measuring effort objectively, like the trap nights for mosquitoes, and you can't identify a good focal group that was sampled similarly to your group. And so in that case, you're left with basically just using the data as they are, doing your best job possible with an M, and finding those clusters and essentially busting them up. I was just talking with John about this, where if those clusters represent clusters of opportunity to detect the species, then you should reduce them. If those clusters are real, so like John is studying a kangaroo rat that has a restricted distribution in Texas, and he assures me that their survey effort was uniform across that distributional area, but they always get more occurrences at the eastern edge. Well, then that may be real, okay? Or, I work from time to time with Ebola and mapping risk for Ebola. And, there we've got like 12 points on the map total. But we do a lot of curation of those points to make sure that each one is a separate independent transfer from whatever the natural reservoir is to a human or to a great ape. And so in that case, I want those concentrations of points. I'm making an assumption that all of those cases that result get reported, which is a dodgy assumption, but it's kind of what we can do.
So on that same line, could you do the target group background, mine is a moth.
Mm-hmm.
I know the species because I look at the whole genus, remove that one species, and make that index map. Would that also possibly show prevalence of density? Because how do you know if you're not living where that species is, whether it's dense in one region or not, and you don't have really good survey data, and you're just grabbing from GBIF?
Yeah. Okay, so one comment is using just that genus may be a little scary because I want the data on the other species to inform about the sampling that could have detected your species.
Right.
And there may not be enough density of species. So you may want to go out to the family, okay, and just take the whole family or whatever the next step up the phylogenetic tree is just so you are not getting into some geographic bias created by,
Yeah, I mean, the UK seems to be a hotspot for finding any kind of butterfly or moth.
or at least for reporting it.
Yeah, exactly.
Yeah.
And that seems to be messing with the results, because that's always a super high.
Right. So again, you can deal with that via some sort of bias surface.
Yeah.
And I would assume, taking all the records of such and such family of moths would show just this huge, solid mass of records covering all of the UK. Or you could essentially take this more informal approach and just say, I'm looking across the range of my species, and across the rest of the range, the density is like, a point every 100 or so kilometers. So I need to reduce the UK to, from 6,000 points to 2. And it hurts to throw away data, but those data are misleading you. You're getting a model more of the UK than of your species. So
Yeah, it's showing sampling effort.
exactly. So what I would do is both, exactly. Other questions about this mess? How are we on time, Mona?
Well, we have to decide if we are doing model calibration and validation next. And I think we should, because a lot of you are interested in that. But that means we're going to hand the paper discussion.
You guys vote.
How many want to leave? We have to get lunch at noon, because otherwise there will be no lunch left. So that's what it looks in the meeting minutes. I wouldn't stretch it past noon.
I can do probably a 15-minute version of this, and that'll leave time for a couple papers. Because Cassandra had one or two that are just superstars there. Yeah.
Just as an aside, I was Googling your paper and the other paper on the Exodes scapularis map.
Yeah.
That you're talking about. Yours is way more accurate than mine, just from collecting around the area.
Thank you. I did not pay the woman, okay?
I will, however, accept that.
Okay, I'm going to go through this model evaluation and model thresholding quickly, but just to kind of repeat some concepts that I think are really crucial for you. Generalities, the calibration data and the evaluation data have to be independent of one another. And when we talk about independence, we're talking about real independence. So let's imagine the typical paradigm, which is I've got 200 points from one source, and so I'm going to randomly split those into 100 for calibration and 100 for evaluation. Right? That's what everybody does. But whatever that source happens to be probably has some biases. Right? I mean, the lazy botanist syndrome is very, very common worldwide, where 95 to 97% of the variation in the position of herbarium sheet records is explained by the distribution of roads. There's also the lazy herpetologist. Birders are better, but anyhow, if there is that bias and you've split your data, then your calibration data and your evaluation data share that same bias. And that means that whatever bias is here, and when you test this model with these data, you're basically confirming the model because those same biases are inherent in both data sets. Many times we don't have any option, but sometimes you do. And when you do, you should take advantage of it. It may be I have, traditionally collected herbarium specimens versus some floristic survey data. It may be that I have, one team collecting one way and another team using standardized trapping, whatever.
If you have like data from like museum records that are verified and data from like iNaturalist or something, would it be better to put the more confident data in the calibration or in the allocation?
It's a good question. No good answer. Because you're also going to have differences in the precision of the georeferencing. The iNaturalist data will tend to be GPSed, and the museum data will tend to be reconstructed from, the textual description of the locality. I guess, again, no good answer. I think you have to think about the merits of both in your system. Sorry to not give you an answer on that one. But anyhow, you need to get your evaluation data as independent of your calibration data as is possible. And essentially what we're doing in this step very narrowly is comparing our model and its ability to predict those independent data to some null model. And, it sounds silly to say only when you have a model that's been tested should you interpret it. But if we were to cruise through the niche modeling literature right now, probably 20 or 30% of the models that are published, interpreted, and explored in papers did not see any testing. And then probably another 40 or 50% of those papers will just give you an AUC score, which is inapplicable to niche modeling. So we have two general types of model evaluations. We have ones that are dependent on a threshold and ones that are independent of thresholds. Okay? So we have two very different views of the world when we look at our map. The threshold-dependent approaches have the big plus of having a very simple test, a very simple interpretation, and very easy computation. What you could call their drawbacks are a set of assumptions that are required for thresholding, and Mona gave you a very good justification of why thresholding should be based on presence data and not on absence data, because we don't have absence data. And the other drawback is that the community tends not to like this. And of course, my response is, I don't care. The other end of the other option is where you don't do a thresholding step. And this has the advantage of avoiding those assumptions related to thresholding. It's very, very well accepted by the community. And probably you could list another one, which would be that you preserve all that information, that this is deep red and this is orange and this is yellow over there, whereas here it's just 1 and 0. Okay? So we're definitely throwing out some information. It may be real information or it may be noise, but we're throwing something out and taking that to binary. Now, the arguments against are that the interpretation is tougher. We know we have huge problems with ROC AUC. The community doesn't care, but they should. And the computation is a bit more difficult. If you read that 2008 paper that Mona and I were our authors on, where we proposed the partial ROC, we did the computations of the areas under the curve by hand, right? So that was a pain in the butt. Yeah.
Has anyone tried a middle path where you threshold, but you don't threshold in a binary way? So you just clip everything below a certain value and then you sort of keep hot spots versus soft spots, but if it's below the threshold, you just go back.
You could do that. or you could have multiple thresholds, which might be useful. Keeping the hotspots is a little dangerous. On the workflow diagram, there was a 2007 paper about thresholding. And what we showed there is that a lot of that variation, between warm spots and hot spots at the upper end of the spectrum, kind of between this and this, a lot of that is related to where you had points for calibration. So it's kind of overfitting at that end of the spectrum. So I'm not super excited about inspecting hot spots versus warm spots for that reason. Yeah.
Would it somehow, doing the continuous, give you a measure, though, of the certainty, or uncertainty, I should say, that you would have on those points, like especially if you're projecting forward? I know what you just said kind of negates a bit of that, but you could say that perhaps you have more confidence in the areas that are the hot spots and have a spectrum.
Well, provided that hot versus warm is real information and not noise. And my experience is that a lot of that upper part of the spectrum variation is related to where did I have greater sampling density, not where is my model more certain.
I'm just trying to figure out, because with binary, then it feels very, like if you're trying to project forward, then it feels like you're not, capturing any of that uncertainty, really.
We didn't go into a huge amount of detail about this, but you remember that we were talking about using lots of different GCMs and lots of different parameterizations. We'll typically look at a variance amongst those forward projections and use that as an index to uncertainty. Okay, those are not the only sources of uncertainty. And I was going to, but decided not to for the purposes of time. I was going to show some other stuff that Marlon has done where he's looking at future climate projections and the sources of variation. So you could have variation owing to RCPs, the climate emission scenario. You could have variation owing to GCMs, the climate model. But he looked at the variation deriving from the different parameter values coming out of the model selection procedure that we showed you yesterday. And the parameter selection contributes about 3 or 4-fold more variation even than RCPs. So it's really scary. It says that we may just be interpreting a lot of noise. And somebody asked about, should we just trash everything that we've done previously? Maybe that was Juan Andres. That one result made me think, oh damn. It's just like all these papers on future climate projections. Okay, binomial tests. I'm going to move fast. We need a thresholded model, so it's just ones and zeros. We need essentially across our test area, and our test area is M. We need what proportion of the area is predicted as suitable. And we would need to know how many test data points we have and how many of them fell within the predicted area. And so just like we can tell who's the better archer, right? We use a binomial test. We have, out of our testing area, the blue area, the red area covers a proportion P. We have a number of successes, in this case 7, and a number of evaluation points, 8. We had 1 failure. And then that's in Excel. I told you the computation was easy. But we're basically just asking, what is the probability of getting 7 or more successful predictions out of 8, given an underlying proportion P identified as suitable by our model? Okay? It's really simple. Can I go on from threshold dependent approaches? Good. Okay. Some of this you've seen before, so I'm not going to spend much time on it. That's the typical rock. And I like to restate these axes. This is avoidance of omission error. And this is proportion of the study area identified as suitable. And so what you can imagine is take an unthresholded maxent output and start with that reddest pixel, right? And that's going to be a tiny, tiny fractional area predicted as suitable, and it's going to leave out almost all of our test points. And so you then just march down the suitability scale, and more area gets added to the predicted area. Everybody with me on that? Until we get all the way down to 0 and the whole area is included. Okay? And we can do some bootstrapping and such to get a variance around that curve and make sure that it is significantly elevated above this. But Mona already told you about why we shouldn't be using RUC. Okay, so I'm not going to. This paper is worth reading by Jorge Lobel, where they list 5 big problems with RUC. Don't need to repeat it. Jorge Soberon, Mona, and I did this paper in 2008, where we basically were saying the same thing as Lobel. We didn't know they were publishing that paper 3 or 4 months before ours came out. But we said much the same things. That's just, the overall paradigm of this is closer to random and that's elevated above random. I've already said much of this. Essentially, the trick that we did in partial ROC, because we're going to move away from this whole square of 1 by 1, because we're going to trim off at least this side of it. So we can no longer say, oh, 0.8 is great, or 0.5 is bad. Because if we come out here, 0.5 may be wonderful, okay? So what we do instead of using kind of the raw area under the curve is for whatever portion of this space we're going to look at, we're going to take the ratio of the observed to the random. That makes sense to everybody? This is random predictive ability. If our curve is sitting down here, if our observed curve is sitting down here, we don't have any predictive ability better than random. So the ratio, it might be 0.51 to 0.5. If that ratio is 1, then our observed curve is not better than random. If it's above 1, then we're better than random. So in this case, we would report an AUC ratio of 1.6. And the beauty of that is now we don't have to use these crude scores, which are meaningful only if the full area is 1.0. So we're going to do things like take our E. Let's see, I've got a better figure of this in a moment, I think. No, I don't. I don't know. There was something on that. But essentially, what we're going to do is, Mona's already explained this to you. We're going to take our error tolerance and use it to pick essentially a point on this axis, and we're going to throw away all of this. And so we're just going to do our area calculations here, having thrown out the part of the curve that has high omission error and that we don't want to interpret. Okay? And so now we're saying, in this part of the curve that is meaningful to us in terms of prediction, does our model perform better than random? And we bootstrap the evaluation points to ask whether, you remember that blue shadow around this line in an earlier one, if that's really big and comes down to here, then we can't reject the null hypothesis, and we say that our prediction is not better than random. Okay? So with partial ROC, you're looking at the ratio, and you're going to count up, as your probability, you're going to count up how many of the random iterations are less than or equal to 1. And if that number is very low, then you're able to reject the null hypothesis. So just to point out, this is this ridiculously cited paper. It's got like 6,000 citations. And yeah, I'm on it, and I hate it. And, but it has this infamous paper. Juan Andres, you'll see soon that this week's movies have this graph, and it talks about the good, the bad, and the ugly. Okay. And did you like the music? So I literally spent hours figuring out how to overlay music on these videos because I wanted every time you see this graph for that theme music from the good, the bad, and the ugly to come out. Exactly. So essentially, ROC approaches were introduced into niche modeling in this paper. That's the ROC AUC, not the partial, the full one. And then this is this weird evaluation approach that they pulled out of a hat for this analysis. There was no published antecedent. It's horribly prevalence dependent, and nobody's ever used it since that I know of. And so this just appeared and then disappeared. And it's really weird that this one paper established this thing of AUC, and now everybody reports it, even though you have two papers saying it's bullshit for niche modeling, but it's there. And because of this, Maxent is up here amongst the chosen, the good. Nobody whistle, please. And then you have these middle algorithms and then the ugly ones. Okay? I'm not going to go into these details. So, again, we've talked about this stuff, and I want to stop at exactly 11:40. So we've talked about this. This is, again, that idea of the size of the area of evaluation makes a difference. And essentially, if you have a predicted area and a small buffer or a big buffer, this is what happens. The proportion covered by the suitable area goes down at every thresholded level, and so that curve kind of migrates up to the left, and then what you have is a higher AUC value. So, back when Mona was in my lab, the joke was, you remember this, oh, your model predictions aren't significantly better than random. Just increase the testing area. You remember that? That was a joke. We didn't do that. But it's true. Back before we had to justify the choice of that area, that's what you could do. Let's stop there.